To start off our video on transcription and gene regulation, we're going to be looking at biological information flow. This is basically the conversion of DNA, which we've always learned is the blueprint of life, into protein, which basically make up everything. They can be structural proteins such as spider silk, they can be catalytic enzymes, and so many other things. We start with DNA, and as we saw in my previous video, the multiplication of DNA is DNA replication. Between DNA and protein, there is an intermediate product called mRNA, which basically exists because it's in a format that can be read to produce protein. The conversion of DNA to mRNA is transcription. Then, the conversion of mRNA to protein is called translation. Now, as you can see, RNA molecules are an intermediate between DNA and protein. Even though the one showed here is mRNA, or messenger RNA, there are two others that play essential roles in this process. One of them is ribosomal RNA, or rRNA, which helps mRNA turn into protein in translation. The third RNA molecule is transfer RNA, or tRNA. This is RNA that has folded into a loop, and it basically connects the language on the RNA to the language of amino acids, which make up proteins. As you can see, we already covered all the learning objectives for 3.1, since we defined biological information flow, the differences between replication, transcription, and translation, RNA molecules, and comparing the RNA molecules. So now on to Unit 3.2, which kind of outlines some nomenclature you need to know for genes involved in transcription. We're going to take a closer look at the DNA molecule that's going to undergo transcription. When looking at the direction of transcription, it's less important in this case to look at the directionality, so 5' to 3' or 3' to 5', and more important to look at the promoter and the terminator. These two are basically sequences that will dictate the starting points and the finishing points of transcription. RNA polymerase, which is the main enzyme that transcribes DNA, will move from the promoter to the terminator. The first base transcribed will be directly after the promoter. And after the terminator is completely transcribed, the enzyme will dissociate and the mRNA will be ready. Since the promoter is not transcribed, and the binding of certain proteins to it can dictate if the gene will be transcribed or not, it's often called a regulatory region, while everything between the promoter and the terminator, including the terminator, are transcribed regions. There are a few sequences within the promoter that are critical to RNA polymerase binding, and they differ between eukaryotes and bacteria. For eukaryotes, it's called the Tata box, while for bacteria, it's called the -35 and -10 region. We're going to talk more about RNA binding to the promoter a bit later. If we look at our learning objectives, we already saw the main transcription units in a gene. We already identified the promoter, and we saw what the promoter looks like in bacteria and eukaryotes. Now we're going to talk about the vocabulary of upstream and downstream. These words are not something you can use in a sentence like, the promoter is upstream. You cannot speak about one region by itself. It's always in relation to another. So for example, you can say gene X is upstream of gene Y, or the -10 region is downstream of the -35 region. We're going to illustrate this with this problem, which is a clicker question from one of Dr. Zeiler's lectures. So the way I like to work through these is that I move my finger or just mentally move my eyes from promoter to terminator and I keep going. So for example, if I want to know what the position is of gene 2 in relation to gene 1, I'm going to start at the promoter of gene 1 and go towards the terminator. So in this case, if I go from promoter 1 to terminator 1 and keep going, I will not encounter gene 2. I will not bump into it. This means that gene 2 is upstream of gene 1. And if you want to know gene 1 in relation to gene 2, you start at the promoter of gene 2 and keep going. So if I go from P2 to T2 and keep going, I will not encounter gene 1, which means it is upstream of gene 2. However, for example, if there were a third gene in this location, and I want to know the position of gene 3 in relation to gene 2, I can start on P2, keep on going towards T2, and if I keep going, I will bump into gene 3. This means that gene 3 is downstream of gene 2. Even though the promoter and terminator are kind of on both strands, RNA polymerase doesn't transcribe both because mRNA is a single-stranded product. The strand that RNA polymerase will transcribe is called the template strand, and since we know that it has to go from promoter to terminator and RNA polymerase works from 3' to 5', in this diagram, the template strand is the bottom strand. Depending on the DNA strand that you were given for a specific problem, the template strand may be the top strand as well. So what you have to do is look from promoter to terminator, which of the two strands follows a 3' to 5' direction. The other strand is called the coding strand. The reason why the coding strand gets its name, at least I personally think so, is because since RNA polymerase is transcribing the template strand, the mRNA product will be complementary to it. And as a result, the transcribed mRNA will end up looking exactly the same, even the same directionality as the coding strand, except that it will have uracils instead of thymines. This is a good trick for practice questions, because if you're asked to transcribe a DNA sequence, you don't have to mentally think about all the complementary base pairs. You can just copy down the coding strand and replace the T's with U's. Now we're going to look at this process a bit more closely in eukaryotes and then in prokaryotes. Within the promoter for eukaryotes, there is the Tata box, which we already spoke about. Here is where transcription factors bind, which increase the likelihood of RNA polymerase being recruited. Though there's multiple, one of them for eukaryotes is the Tata binding protein. So when these transcription factors are bound, RNA polymerase is recruited and begins transcribing from the promoter to the terminator. Even though it binds to the promoter, the sequence that makes up the promoter is not part of the mRNA transcript. The DNA molecule here actually represents both strands, so the directionality I put of 5' to 3' is just meant to represent the directionality of the resulting immature mRNA. In eukaryotes, the mRNA produced from transcription is actually not ready for translation at all, since it is composed of introns and exons, where exons are parts that code for proteins, but introns are regions that are non-coding. Since a cell doesn't really want to waste energy transcribing regions that won't turn into a protein, the introns are removed by a process called splicing. Splicing results in some interesting scenarios, since even though all introns are removed, you can remove different combinations of exons. So for example, if you remove no exons, here you would be removing only the introns and no exons, resulting in an mRNA that will be made into a specific protein product. However, this protein product would be different if, as well as the introns, you would remove exon 1 or exon 2. After splicing, the mRNA is still not ready for translation. This is because of the location of transcription versus translation in eukaryotes. This process is happening in the nucleus. However, rRNA and ribosomes for translation are present in the cytoplasm. So this immature mRNA has to be transported to the cytoplasm, for which it needs some protection on its 5' and 3' ends. Hence, there is a 5' cap added to the 5' end, and what is called a poly-A tail added to the 3' end. Now, this eukaryotic mRNA is ready for translation. Now we're going to move on to the same process in prokaryotes. So if we remember the Tata box from eukaryotes, in prokaryotes, this is called the -35 box and the -10 box. So this is where the transcription factors are going to bind. Specifically, a protein called a sigma factor will bind to the promoter and recruit RNA polymerase. I have noted here that the first base that is transcribed in prokaryotes is called the +1 site. This information is useful when completing worksheets or being asked to transcribe a DNA sequence because the +1 site indicates from where you need to start transcribing. For prokaryotes, the mRNA that is transcribed is already the mature mRNA. It doesn't require modification because in prokaryotes, transcription and translation happen in the same place, since there is no compartmentalization of organelles. Since the mRNA doesn't need to be transported, it doesn't need to be modified with protection. Furthermore, prokaryotic mRNA is only made up of exons, so no splicing needs to happen. The fact that there are no compartments and no membrane-bound organelles also means that transcription and translation can happen simultaneously. We have hit all the learning objectives, so now we're going to move on to Unit 3.6 Operons. Usually, we already know that the structure of a gene that's going to be transcribed is basically a promoter, a terminator, and one gene in the middle. However, prokaryotes and some eukaryotes have many genes between the promoter and the terminator. This means that they are all transcribed together into one mRNA. However, after, they are translated into different proteins. The expression of these genes is dictated by an operator, which is kind of on or around the promoter. A molecule will bind to it, which will either repress or activate transcription. These two modes of controlling whether or not transcription will happen at a high level are called positive and negative regulation. In positive regulation, the promoter in the operon is kind of weak, which means that it can't recruit RNA polymerase very efficiently, resulting in low or basal levels of transcription. Note that transcriptional level is almost never zero. We call it basal because even when it seems like there can be no transcription, there must be extremely low levels to allow for the regulatory molecule to come inside the cell. Since the promoter is weak in positive regulation, it kind of needs an extra help to recruit RNA polymerase, which is where the activator comes in, which is a molecule that binds to the operator and increases the chances that the promoter will recruit RNA polymerase. In negative regulation, there's already a regulatory protein bound to the operator, which is the repressor. This molecule prevents the promoter from recruiting RNA polymerase, resulting in low or basal levels of transcription. This happens because the promoter is very strong, so without the repressor, the gene would be always expressed, which you don't necessarily want. As an example of positive regulation, we're going to look at the male operon. In this diagram, you can see two sets of promoters and terminators. The one on the right, as you can see, is attached to an operator, which is that blue shape with an O, and in the middle are two genes that are MALP and MALQ, which, when translated into protein, turn into the proteins MALP and MALQ, which break down maltose, which is a carbohydrate that, when broken down, provides energy for the cell. In this case, the operon is made of P2, the operator, MALP, MALQ, and T2. P2 is actually quite a weak promoter, so it doesn't do a very good job of recruiting RNA polymerase to initiate transcription. This is actually something quite convenient because you really don't want your cell to be wasting a lot of energy in producing MALP and MALQ, the proteins, when there's no maltose to break down. So ideally, our promoter will need some help from an activator to be able to recruit RNA polymerase, and will ideally only do so in the presence of maltose. So this gene segment on the left is like any other gene segment because it's not affiliated to an operator. It codes for the protein MALT, which acts as an activator by binding to the operon. However, in its normal state, it doesn't have a really high binding affinity to the operator, so it doesn't bind to the operator. However, in the presence of maltose, a conformational change happens that makes the MALT have a higher binding affinity for the MAL operator. The binding of the activator to the operator increases the chances that RNA polymerase will be recruited. Therefore, in the presence of maltose, the genes in the MAL operon will be expressed. Without maltose, the activator can't bind to the operator. Therefore, the genes in the operon will not be expressed. Now we're going to look at our example of negative regulation, which is the lack operon. Like with the mal operon, there's two pairs of promoters and terminators. The one on the right is what is the lack operon, and it has three genes, LAC-Z, LAC-Y, and LAC-A, which each code for a protein that breaks down lactose. The gene segment on the left, which is the normal gene segment, codes for LAC-I, which is a repressor. This repressor is constantly bound to the operator, such that the promoter, which is a strong promoter, cannot recruit RNA polymerase. So in the normal state, in the absence of lactose, no transcription will occur because the repressor is bound to the operator. However, lactose binds to the operator and moves it away from the operator, hence allowing the strong promoter to recruit RNA polymerase and transcribe the genes. Therefore, like with the mal operon, without the presence of the nutrient, there will be no transcription. However, when lactose arrives, since it moves the repressor away from the operator, P2, which is a strong promoter, can recruit RNA polymerase and transcription will occur. Now we're going to compare what we've learned about the lac and the mal operon in relation to gene regulation. As we already established, the lac operon is an example of negative regulation and the mal operon is an example of positive regulation. The lac operon has a strong promoter, therefore it needs a repressor to prevent this promoter from constantly expressing these genes at a very high level. For the mal operon, its promoter is very weak, so it needs a little extra help by binding to an activator. In both cases, transcription is very low without the nutrient and is high with the nutrient. In the example of the lac operon, it's low because the repressor is bound to the operator, preventing the promoter from doing its job. And in the mal operon, it's because the promoter is very weak and does not have the ability to recruit RNA polymerase. Lactose binds to the repressor and moves it away from the operon, while maltose binds to the activator and moves it to the operon to help with transcription. Therefore, in the absence of the nutrient, in the case of the lac operon, the repressor is bound to the operator, while in the mal operon, the operator stands alone. And in the presence of the nutrient, since lactose has removed the repressor, the operator stands by itself, while in the case of the mal operon, since maltose has moved the activator to the operator is bound to the activator in the presence of maltose. So we already talked about how a nutrient like lactose or maltose can affect the regulation of an operon. We drew an operon. We spoke about strong and weak promoters, also about positive versus negative regulators and their binding to the operator, and signal molecules in positive regulation. Just for reference and some vocabulary, an inducer refers to a molecule that regulates gene expression. Now we're going to go through a few clicker questions relating to mutations within the lac and mal operons. We're going over them because these are clicker questions that many people had questions about in class. I think to answer these questions, the best way to tackle them is to draw what the gene organization looks like in the normal cell versus in the mutation. So this question is asking the scenario where the operator is removed. So compared to a wild-type cell, what will the mutant phenotype be in the presence of lactose? So if we draw the normal cell, the operator still exists, and since lactose is there, it binds to the repressor and removes the repressor from the operator, allowing for RNA polymerase to bind. So transcription level is high. The mutant does not have an operator, making these genes basically normal genes. So in a normal gene, since there is a promoter and a terminator, transcription will happen normally. So we can also say that it happens at a high level. Therefore, the mutant roughly has the equal amount of LAC-Z production than the wild type cell, as we can see here. So the answer is C. This next question has the exact same scenario, only that in this case, there is no lactose. So the wild type cell will have an operator, and as we said before, in positively regulated genes, the repressor is bound to the operator in a normal cell where there is no nutrient. Therefore, RNA polymerase will not be able to bind, so transcription will be low. The mutant doesn't have an operator, which, as we saw before, means that it's just a regular gene that is transcribed normally, so we can consider its transcription level as high. Therefore, in this case, the mutant will have more LAC-Z production than in the wild type. This next question is removing 3 elements, which is the terminator of LAC-I, the promoter of the operon, and the operator. So the wild type cell will have all of the normal components, and in this case, lactose is present. This means that lactose has bound to the repressor and removed it from the operator, allowing RNA polymerase to bind and transcription to happen. So transcription levels are high. In the mutant, there is 1 promoter and 1 terminator for all of these genes, when originally each one had their own. This means that when RNA polymerase binds to the LAC-I promoter, which it can do because now it's just a regular gene since it has no operator, it will go all the way to the terminator of the operon. The way I like to think about this is that since it wasn't meant to go that far along, it kind of does a mediocre job, so transcription is kind of medium or low. Therefore, the mutant will have less LAC-Z production than the wild type. Now we're asking the same question, but in the absence of lactose. So in the wild type, without lactose, the repressor will be bound to the operator, so RNA polymerase cannot bind. So transcription will be low. In the mutant, as we said before, this gene will turn into a regular gene since the operon is removed. However, since it's using the promoter from LAC-I, transcription will not be as strong since it kind of wasn't made to go such a large distance. So transcription will be medium. However, this is still larger than the amount of transcription that happens in the wild type. Therefore, the mutant will have more LAC-Z production than the wild type. These are all the mutation questions pertaining to the LAC operon, so now we will go on to the mal operon. In this scenario, the promoter of mal-T is removed. So in the wild type, everything will be normal, and if we remember from what we said about maltose, there is nothing that is originally bound to the operator, it stands alone, but the promoter is very weak. So it kind of needs a little push from the activator. However, the activator can only bind to the operator when maltose is present. In this case, maltose is absent, therefore RNA polymerase cannot bind and transcription will be low. In the mutant, since there is no promoter for mal-T cannot be produced, since there is literally nowhere for RNA polymerase to recognize that it should start transcribing there. Since there is no activator, and either way, if there were, as we see with the wild type, it could not bind to the operator because there's no maltose, there will also be low levels of transcription. Therefore, the mutant and the wild type will have roughly equal levels of mal-P production. Now we have finally covered the last learning objective in the unit pertaining to mutations in operons.